All right, today we're going to primarily be looking at genetic mutations, but we'll also be looking at how to read and analyze a codon chart. All right, so if you remember last class, what we talked about is replication and then transcription. In order to read a codon chart, you're going to have to transcribe a DNA sequence, so you'll need to have the mRNA sequence. Please remember the mRNA sequence is known as the codon, so it's the codon sequence or the codon mRNA sequence. So basically what it is, if you noticed, we also do everything in sets of three, and that's going to help out a lot. So we've already went over this last class, but just in case you don't have the answers, if you will, write down this mRNA sequence, and I'll go ahead and go through it real quick. So remember, G pairs with C, C with G, so the next letter should be G. And this is how we're reading it. We're only reading here. In the past, students have tried to read it across the top from A, T, A. But whenever we look at a codon chart, you'll see that you can't have any T's. The T's would have to be a U instead. So we're only reading the column that's for the codon. So again, I'll start over. So this one right here would be G, C, G, T, A, U, A, T, A, G, C, G, C, G, C, T, A, U, A, T, A, T, A, C, G, C. Now that you have this whole column here, what I would do is I would circle them in threes. So the first three would be the A, U, G, and just keep doing that. That'll help organize your information together. And once you have that, we'll go ahead and flip over here to the next slide. So this is a codon wheel. So I know this may look really daunting, but it really won't be that bad. So remember, those first three letters are A, U, G. The next three letters were G. Having to write this down myself real quick. T, A, U, A, T, A, G, C, G, C, T, U, A, C. All right. Oh, C. All right, so whenever you read this codon chart, we're going to look at the first three. The first three was A, U, G. So based on the information that I gave you on it, you always want to start in the center of any target. You're always aiming for the center first. And here's that A right here. And then the next letter is U. Now we're not going to this U. Now we want to move out to the second ring. The second ring, we're looking for the U that's connected to this A. So the next U would be right here. We just drop down A, U, and then we're looking for the next ring for the third letter, and that would be G. So it goes A, U, G, and if you notice, it has that little triangle there where it says M-E-T. Well, and on another one we'll see, because a lot of these are put into three-letter increments, but we can actually see some of the names of them on the next one. So that would be M-E-T. But based on this legend we have over here, this triangle means start. So that's our start codon. So what that basically means is if you think about a sentence, every sentence starts with a capitalized letter. So that means every gene should start with the start codon, which is that one. If you notice, the black circles mean stop. And these are the stop codons. So think about how you end a sentence. You end a sentence with a period, a question mark, an exclamation point. Well, this right here has three stop codons, and so that's how you end the gene. Okay? And anytime you have a mix-up right there that causes them to not stop or, not start correctly or any of those mishaps, well, that would be a genetic mutation. So let's go ahead and look at the next three. The next three codon letters were G, U, A. So again, let's start here in the middle, G, and then we go out here to the green one on the next level, that'd be U, and then we'd go out to A, and that's right here, and that leads us to VAL. Now, did you notice that all four of these, whether it's GUG, GUA, GUC, or GUU, all of these would all give us the exact same amino acid. They all would. And that's going to be really important whenever we talk about in the next, our next little bit of information, our next section of information about genetic mutations. Because there's some genetic mutations that it doesn't matter. Because let's say there's a mutation here where instead of having an A, we had a C instead, and it was GUC. Well, we would get the same exact thing, so it really wouldn't matter genetically speaking. That happened. That mutation really wouldn't affect anything, even though there was a mistake there. So if you would, go ahead and try the next one on your own where it says GCU. Try to do GCU on your own. All right, I'm about to show you, so if you're still working on it, please pause the video. Here's G, here's C, here's U, and that would be the amino acid ALA. And that's just the abbreviation. They typically all have, INE on the end of it and things like that. So that'd be ALA. And then the last one would be AAC. So pause the video so that you can do it. And I'm going to go ahead and do it now. So here's our A, here's our second A, and then here's our C. And so that amino acid would be ASN. And what this is each gene's about 120 nucleotides long. So you'd end up having to, basically do this another 40 times or 36 times to figure out the gene, the amino acid sequence. And of course, it should end with one of these up here. Of course, that's the stop. I should be putting the period at the end of it. So this is another type of codon chart. This might look a lot more confusing now, but it's actually not too bad. If you notice right here where it says first base in codon, so that means the first letter. Up here at the top it says second base, that would be the second letter, and over here on the side it says third. So what you have to do is you have to first find out the row that it's on, and then you have to find out the column, and then you have to find out the specific letter, the specific one that it's on. So let's look at AUG again, those first three letters again. So the first letter is A, so that means I know that the amino acid I'm looking for is somewhere on this A row, and that's this long row going all the way across the top. And after that, then I go to the next letter, which is U, so I know my answer is somewhere right up in here. Oh, I was hoping I would highlight, but it didn't. So then I go to the second letter, which is a U, and that tells me that one of these four letters, whether it's ISO or MET, in which these are the same ones at the top, but one of these four columns, or one of these four amino acids is my answer. And the last letter is G. Here's the G, I come across, and boom, now I know that it's methionine. And so that would be my start codon. We'll go ahead and look at one more on this one. GUA, so this is G. So that means I know that my answer is somewhere on this column in this G row. And then the next letter is U. So I come all the way over here to the U. Now look at this. You see how all four of them are the valine? They're all the same exact thing. Well, that means I don't even care what the third one is because I know the answer is going to be valine, period. I already know it is. Just like if the answer was GC and I see alanine, then boom, I know that it's going to be that answer. All right. And we'll be practicing with both. And the reason why I give you both, because even though this circle one, once you know how to use it, this wheel one is really fast and easy to read. You don't ever know which one you're going to be given on a test, or especially on your state exam. And so I like to teach you both of them just in case. And remember, we're using the mRNA codon to read this. You're not able to use the DNA because if you notice there's no T Instead of the T, it's the U. And if you use the tRNA to read this, then every answer you get is going to be wrong. So make sure you use the mRNA sequence. All right, so what we're going to learn about next are the different types of mutations, genetic mutations. We need to make sure that we're able to explain each type and understand the difference between a point mutation and a frame shift, and then be able to analyze an image of a genetic sequence and this specific mutation. So basically you have two general types. You have a point mutation and a frame shift. So a point mutation is nothing more than a substitution. A point mutation only changes one specific point on the DNA sequence. Whereas a frame shift, wherever that mutation is, not only does it affect that point, but it affects everything after it as well. And we have two different types here. There's actually more than this, but these are the ones that we're going to be focusing on. We have deletion and insertion. Sometimes you'll see it as addition. And of course, point mutation would be substitution. So substitution occurs whenever the base pair is replaced with the wrong base pair. So this is something that I started doing a couple years ago. Instead of using the letters, the A, the T, the C, the G, or the A, U, C, G, instead of looking at the letters on the DNA and RNA strands, I started doing it with numbers, and students caught on to the idea of what was happening so much faster. So a normal sequence should go 1, 2, 3, 4, 5, 6, 7, 8, 9. But if you look at this, now it's 1, 2, 3, 6, 5, 6, 7, 8, 9. Well, I substituted that 4 for a 6. You see how it only changed this one thing? It didn't change the 1, 2, 3. It also didn't change the 7, 8, 9. Now, remember also, the reason why I have these grouped in threes is because that's how you read your codons for your amino acid. That means this is the only amino acid that's going to change. The first one's still going to be the same. The third one's still going to be the same. The rest of the line, the other 37 of the amino acids are still going to be the same. This one's going to be the only one that's different. So a silent mutation, this occurs whenever we replace something. It actually gets replaced with the same amino acid. That's what I was talking about earlier on that other slide. Now, on a disaster, anything like that would happen. So if you remember, I talked about a stop codon would end the sequence. So basically, if I was to end a sentence early and put a period there, that would, basically destroy that sentence. And so that's what a disaster mutation on a substitution would be. Typically, your silent mutations, everybody has these, just because all that is just swapping up a letter and you end up with the same exact amino acid. So this is substitution, and substitution is a point mutation. So here's an actual example of it. So over here we see this normal gene. We have G pairing with C, G, C, T, A, CG, TA, CG. We have all of this, and it's replicating normally, no problem. Transcribing normally. We get our regular letters here. No problem there. But over here, on the other hand, now, one thing I do want to make sure that you do understand is what we're doing right here, this first level coming down, this is us replicating it. So whenever we talk about these genetic mutations, we're talking about an issue in the replication process. I just had somebody ask me that question, so that's why I wanted to go over that real quick. So right here, basically what happened here with this A got misreplicated, which should have been a T. And then we came down here and we had a U here. Well, that part was done correctly, but getting that T there was the wrong part. It got changed. It got substituted for the wrong letter. Well, that caused this amino acid to not be the correct amino acid. Insertion and deletion, or insertion and addition. This is a frame shift that's going to affect the entire line, and this is where we insert an amino acid that should not have been there at all. So over here on the left side, we see our normal gene set up, but over here on the right side, things have changed. So they've inserted a guanine, and that caused this chain to instead read CCAG. Now it reads CCACGA, but you notice that it keeps moving every letter on down the chain. And whenever it does that, that means the rest of this chain is going to change. So that means if it started here, then that means every single one of these is different. That's the reason why you have more issues at a frame shift. And that's a good way of remembering how it shifts the entire frame all the way down the chain. Same way with deletion. Whenever you delete a letter, that means that everything had to move in. And again, it changes the whole chain. So I found this image and I really liked it. So basically, if this is supposed to be our normal one right here, where it says 9 bases and it comes over, and we end up having a substitution, so we take out the C and we put an A in its place, that means it changed that one letter. But if we came up here and we just put an A in, that would be insertion, so that means now we have extra letters. Or if we removed a letter, then that means that it's going to squeeze down and only going to have 8 bases. So this is typically a tip that I give students. If I'm asking you to look at a DNA strand and try to find the genetic mutation or what is the genetic mutation here, the easiest way to do it is just to count it. If you look at the original, the normal one, and I tell you that there's 9 there, and in the one I'm asking about is 10, then that means I added 1, right? So that means it would be insertion. If the normal one has 9 and then the next one has 9 as well, we know that it's substitution because all we did is replace. Deletion, if it has less than the original, then that means it's deletion. So that's a little tip there for you to be able to kind of navigate that and try to pick them out, because sometimes it can be a little bit daunting. And so hopefully that tip helps out. So as far as your review, like right here, this is what I mean. So on the left side, I give you the normal ones, but then on the right side, I'm asking you what it is. So of course the normal side is 9 letters, but if you notice right here, it's still 9 letters, but there's a mistake, so that would be substitution. 9 letters, oh, well this right here is only 8 letters, so that's deletion. 9 letters, oh, this one's 10 letters, so I know that's insertion. So make sure you can do that type of thing. And also don't forget you'll need to be able to describe it as a point shift or a frame shift. So since this one's 9 and 9, I know that this one's point. This one's 9 and 8, so that's going to be a frame shift. This one's 9 and 10, so that's a frame shift as well. All right, and that is it. And that's it for this unit as well. So don't forget, be looking at your quizzes, go through that, be looking at the work that we did on this section, and make sure you come prepared for this test.